He has to date published over 250 peer-reviewed papers, and he was listed as a Clarivate Highly Cited Researcher for 2023. His research output and his achievements have been recognized through national and international awards, a fellowship, including the 2017 Australian Research Council Future Fellowship. He is editor of the Journal of Hazardous Materials and associate editor for Water Research. Please welcome to the stage Professor Guo.
Thank you very much for your kind introduction, Jeffrey. And also, it's a truly pleasure to be here. And actually, this is my fourth EDA conference, since first one in Michigan 2017, I remember. So today, I would share our research in the last 10 years. So my topic will be focused on evolution, emergence, and transmission of AMR associated with non-antibiotic pharmaceuticals. So here I also give a subtitle. So visualizing the invisible. So what does that mean? So I hope once I finish my talk, I can offer you a clear answer eventually. Okay, so I designed the 9 questions for my talk. I'll go through one by one. So first question I want to ask, so how does AMR emerge and spread? Actually, for the ARB or ARGs, so they don't need a passport. They don't need a visa even. So they can transport or transfer from Australia to Canada, from Canada to Europe to Africa. So that's why, in order to address AMR, so we need a one health agenda or approach. So, but from the microbiology point of view, so there are 3 major pathways to cause the AMR transmission. First pathway we call the mutation. So if we have a sensitive bacteria like E. coli, for example, so if we pose like a sensitive bacteria to antibiotics, so antibiotic actually is a selective or stress pressure. So they can trigger mutation, or they can trigger SOS response. Also, they can trigger RS overgeneration. So after that, so they cause the DNA damage. So then some genetic mutation will be happening. So bacteria can't respect this like a mutation. As a consequence, sensitive bacteria will become antibiotic-resistant bacteria. So this is a mutation. Second pathway is a VGT, vertical gene transfer. So this is very straightforward to understand. So if a parent cell carries some antibiotic-resistant gene, ARG, in their genome, actually this parent cell can pass their ARG to the daughter cells generation by generation. So this is a VGT. Third one is much more complicated. So we call the horizontal gene transfer, showed as HGT. So actually including like 3 cyber pathways. So let me explain one by one. For the first one, conjugation. So if we have donor cell, if they carry some mobile genetic elements, such plasmid, so we also have a sensitive receptor bacteria. If we mix them together, actually they can build up the physical contact, like the bridge. So then mobile genetic elements such plasmid can transfer from donor to recipient. So this recipient can integrate that plasmid in their genome. As a consequence, sensitive recipient will become ARB. So this is conjugation. So second pathway is a transformation. So transformation means under certain conditions, for example, if we have antibiotics, they cause cyanosis, some DNA will be released. So we have free-living DNA, but the free-living DNA even can carry some ARG on it. So then, for the competing cells, they can swallow or uptake that free-living DNA. Then they can integrate that ARG in their genome. So then, sensitive recipient will become multiple drug-resistant bacteria. So this transformation. The last one is transduction. It's a very unique pathway because so that process is mediated by bacteriophage. So bacteriophage, like a shuttle bus, they can carry antibiotic-resistant genes from the donor, then they transport to the recipient and inject that ARG into the recipient cells. As a consequence, recipient can integrate that ARG, become multi-drug-resistant bacteria. So here, we have 3 cyber pathway: conjugation, transformation, and transduction. Okay, so this is a very basic knowledge. So question we want to ask is, so as we know, the misuse or overuse antibiotics has spin up the gene transfer or mutation, trigger the AMR transmission. But the question here we want to ask is, are there any other invisible triggering factors? Okay, so what we want to address is how about non-antibiotic pharmaceuticals? So in drug market, no human targeted drugs actually account for 95% in the drug market. Antibiotic just 5%. Is there any relationship between the non-antibiotic pharmaceutical and antibiotic resistance? So this is a question no one addressed before. So that's why we did some experiment, try to demonstrate or prove this is happening or not, and the interaction. Okay, the first model we did is a conjugation model. So we choose E. coli with RP4. For that RP4, they carry 3 antibiotics, ampicillin, tetracycline, and clindamycin. So that plasmid carried by the E. coli bacteria. We choose 2 receptor bacteria. One is E. coli, and one is Pseudomonas petita. So we mix them. So you can imagine that plasmid can transfer from donor to recipient. So after that, that recipient bacteria will become Tres conjugates. So, but how to select the Tres conjugates? So how to quantify Tres conjugates? We can use antibiotic like a selective place. We add 4 antibiotics for that place. So now you can imagine donor count group, because donor is very sensitive to CHL antibiotics. Recipient doesn't grow either because recipient is very sensitive to other 3 antibiotics. Only Tres conjugates can grow on that selected place. So then, based on this equation, we normalize the number of the Tres conjugates to the total number of the recipient, so we can get Tres conjugation frequency. So this is so the question now we want to ask is, spontaneously for the gene transfer from donor to recipient is very, very limited. 1 million bacteria, only very few can acquire that plasmid. But now, once we add no antibody pharmaceutical, so how about that process? Will it be promoted or will it be suppressed? So this is the question. Okay, so first chemical or drug we test is carbamazepine. So this drug definitely is not antibiotics. It's for the anti-epileptic drug. It's very essential, human targeted drug. So actually, this drug is also very, very robust, can't be degraded in the environment. So that's why we can detect the carbamazepine in our groundwater, surface water, even potable water. So up to the milligram per liter. Okay, so then we add this carbamazepine to our gene transfer model, conjugation model. You can see here. So this is the control without any carbamazepine. So this is 0.05 milligram per liter. So then we increase concentration. You can see for the control, just very few Tres conjugates. But once you increase the carbamazepine dosage, you have more and more Tres conjugates popping up on that selective plate. So this means, indeed, carbamazepine is not antibiotics, but indeed it can promote gene transfer. So how about the photo change? We did a statistical analysis. So blue bar shows from E. coli to E. coli, and red bar shows E. coli to Pseudomonas petita. So you can see under exposure of these drugs, so gene transfer frequency has been significantly increased. It also shows dose-dependent trend. So increased concentration, high frequency. Okay, so this is only phenomenon. So why this is happening? So first idea we come up is how about the radical? How about the reactive oxygen species? So that's why we quantify the ROS generation in donor and recipient bacterial cells. You can see here, for the control, no overgeneration. But once we add carbamazepine, so no matter for donor or to recipient bacteria, they do overgenerate radical, right? So this, you can say, we have enhanced the gene transfer, we have promoted RS generation, but this is just a correlation. Is that is causation or not? We can't say that. So that's why we did another experiment. We add RS scavenger. So RS scavenger, actually, they can quench, they can clean off radical. So then we redid that a gene transfer experiment again. So now you can say, for that gray bar here is without radical, without the scavenger. So then for the bluebird, we add a radical. You can say after adding radical scavenger, the gene transfer frequency drop back to the control level. That further confirm RS overgeneration is a very, very important the reason for the gene transfer enhancement. Okay, so after that, we did whole genome RNA sequencing and protein sequencing. We also confirm for the bacteria, like the donor or recipient, some RS-related gene, for example, SOD gene, have been upregulated because bacteria they want adapted stress. That's why they try to upregulate their response gene and reduce the stress. Okay, this is the first mechanism or pathway. The second reason, how about cell membrane permeability? Because for the gene transfer, they have to overcome that cell membrane barrier. So that's why we try to say maybe cell membrane permeability has been enhanced. So that's why we did the TEM imaging. You can say under exposure this drug, some cell membrane has been damaged. So we also did a quantification based on the flow cytometry. So under this drug exposure, no matter for the donor or recipient, cell membrane permeability also has been enhanced. Again, we run RNA sequencing and protein sequencing. Further confirm some membrane-related gene, such as OMP-related protein or gene, has been dramatically upregulated. Okay, so together with, we propose this pathway. Under exposure carbamazepine, actually, they can induce RS overgeneration. They can trigger SOS response. They can enhance cell membrane permeability, and also they can regulate the pallets related gene. And then, as a consequence, the plasmid will be easily transferred from donor to recipient. Okay, so this is the first study we did. It's kind of proof of concept. But the next question will become, okay, this phenomenon is universal or is drug specific? How about other drugs? Okay, so this time we choose 6 different drugs, including 3 anti-inflammatory drugs. They're very commonly used. And we also have 1 lipid lowering drug. We have 1 beta blocker and 1 contrast media. So for all of these 5 drugs, except last one, for the media, contrast media, it will not enhance gene transfer. But for the other 5 drugs, they do enhance the gene transfer. You can see for the purple bar is the last one, but for the other 5 drugs, no matter which drug we have, they can induce gene transfer enhancement. So why is this happening? So we did we compared the 2 category drugs. For the top line is non-antibiotic pharmaceuticals. Bottom line is commonly used antibiotics. You can see actually they share very similar function groups. They do have a capsule function groups. So that's why our hypothesis here is maybe some drugs, human target drugs, they also can have antibiotic-like roles because they can trigger the oxidative stress. So, but we can't do the experiment from A to A, B, C, D, E, etc. So that's why currently we try to use a machine learning model to predict which drug or which chemical can have this antibiotic side effects. So this is an ongoing project. Okay, now we come to a fourth question. So how about the transformation? I mentioned under certain pressure, DNA will be released. We have a free DNA. That DNA can be uptaken by the competing cells. So then we build up a second model we call the transformation model. What we choose is we choose a PWH plasmid and Acinetobacter as a competing bacterial cell. So we mix them together. Spontaneously, that plasmid uptaking also very, very limited. Just very few cells per million can uptake that plasmid. But now, we want to test under exposure these drugs, so process will be increased or decreased. So we use a similar method to quantify transformates, and we also did RNA and protein sequencing to try to understand why this is happening. Okay, let's have a look at results. So again, we choose these 6 drugs, including anti-inflammatory drugs, lipid drug, and the beta blocker. Last one is like a contrast media. It will not increase too much, but for the other 5 drugs, they can increase gene transfer through the transformation. We also have a dose-dependent trend for these 5 drugs. Okay, so this is what we did. We developed conjugation and transfer model to confirm some drugs, they are not antibiotics, but they can promote the gene transfer. But for the microbiology experiment, you will be challenging me. Seeing is believing. Do you have evidence to show that process? Can we visualize that process? Okay, one of the PhD students developed a new platform. So we integrate the microfluidic system. We use fluorescent bacteria. So then we put a place under the confocal microscopy. We want real-time monitor that process. Okay, so let's have a look at it. Okay, the red here are donor, black one are recipient. Once recipient uptake that plasmid, they will become green fluorescence. So you can say without any drug dosage, that process is happening, but very, very slow. After 2 hours, under this field, only very few bacteria become trace conjugates, right? So this is control. Let's move to the second one, is treated group. So we have a donor as red, and black here are recipient. You can see after half an hour, we have more and more trace conjugates pop up. Much more trace conjugate bacteria comes in control. So this, once again, using such platform, we confirm no antibiotic pharmaceutical, they can promote gene transfer. We can visualize that process. Okay, so 6th question. So I did like a horizontal gene transfer. So how about the mutation? So can these drugs trigger mutation or not? We build a mutation or evolution model. So this time, we choose 6 anti-dependent drugs. This is for the treatment like mental health, for the depression disease treatment. So we choose very commonly used drugs. So then we did like a evolution or mutation experiment. So we choose sensitive wild type E. coli. We do the passage day by day. So every day we passage like 1% bacteria to next tube until like the 2 months. So then we collected a time course sample to confirm. So we add antibody for that place. For each antibody plate, we just add an individual antibody. If some bacteria clone can grow, that means they are mutant. Okay, we did that. We can use this equation to quantify mutation frequency. So after that, we also randomly pick up some cloning. We scan the MAC against multiple antibiotics to confirm are they acquiring multiple drug resistance or not. So we did such an experiment. So let's have a look at the results. We choose such as sertraline as an example. This is a very commonly used anti-dependent drug. So what you can see here, so we dose 0.1 ppm until like 15 ppm. So high concentration, the quicker mutation happening. So even for the 0.1 ppm, after 1 month or 2 months, they do enhance like gene transfer frequency dramatically. So then we pick up some cloning, scan the MAC. Actually, you can say here, so they develop multiple drug resistance rather than just individual resistance. So this is a mutation model. Why is happening? Because under exposure to this drug, so we do have some SNP. We have a genetic mutation like happening in the bacterial genome. So we run the whole genome DNA sequencing. We can pick up some like SNP like in the genome. So here, I don't want to go through the details. So but we do have some evidence. So another reason is actually for the E-plus palm under such exposure, that E-plus palm has been upregulated. So that's why, so this bacteria not only resistant one bacteria, but become multiple drug resistant. They can pump up multiple drug like antibiotics. Okay, so this is all the experiment, no matter gene transfer or mutation, is related like a planktonic cell or pure culture cells. So how about the community? And in the wastewater treatment plant or the heme gaunt, So this is happening a lot. So we did a further experiment. Okay, so this time we pick up like an active sludge as our recipient. It's a mixed control. It's a very diverse like a bacterial community. We choose like a E. coli with a PKGK5 plasmid. So we add them together. So now you can imagine, so some active bacteria will acquire that plasmid. But how could you identify which bacteria become trace conjugates? So that's why we can do a cell sorting. So we can sort out like the black cell, red cell, the gray cell. So then with that gray cell, that means trace conjugates. Then we can run the 16S sequencing. We can identify like who become the trace conjugates. So again, we can use them like a carbamazepine as an example. You can say under exposure carbamazepine, we do have a dose-dependent increased threat. And we also confirm like which bacteria become, even for some like waterborne pathogen, Legionella, they can acquire that plasmid. Okay, so this is a 7 question. Now this second last question. I'll try my best to finish in 1 minute. So how about human gut, right? Because patients uptake that drug very regularly. So once we uptake the drug, can they change microbiota or gut microbiota or not? Can they promote gene transfer or not? So this is still a question. That's why we did animal experiment. We collect the fecal sample from mouse. We mix that donor with fecal sample. We did this experiment again. We use a very similar method, cell sorting plus applicant sequencing. We can confirm who become trace conjugates. So answer is yes. You can say for this like a anti-dependent drug, they can promote gene transfer in the animal gut. So we also did the 16S RNA sequencing. We can find some pathogenic bacteria can acquire that plasmid. Okay, so what does that mean? Maybe this gut can reshape your gut microbiota, can promote so gene transfer. But if you are interested in the details, you can look through our publications. Okay, last question. We challenge ourselves. What else? What should we do further? So currently, we develop animals. We do the in situ like experiment. We garbage like a bacteria where we feed drug. So we want to test how about IBD. Probably they can cause some disease. So this is an ongoing experiment. So with this, probably I do a very short summary. No antibody pharmaceuticals, some like human targeted drugs. They are not antibiotics, but they can trigger mutation. They can promote gene transfer. So we build up multiple models, including conjugation model, transformation, mutation, community level. We confirm they do have such side effects. This is like iceberg. So they are not antibiotics, but they are invisible factor, can trigger AMR transmission. So lastly, I would acknowledge all the team members. They work very hard in the lab. And also I would like to acknowledge my funding agency and industry partners. Thank you for your listening.
Yes. Joachim Larsson, University of Gothenburg. So you just started in one of your first slides talking about mutation, and you introduced antibiotics as something that induces mutations. Mm-hmm. But I guess the most important part of antibiotics in this is the selection by antibiotics of the mutants. Mutations occurs all the time anyway without the presence of, and it can be basically enhanced by all types of stresses, probably hundreds of thousands or even millions of different chemicals, and has done so for eons. Same thing with horizontal gene transfer. Mm-hmm. Lots and lots of things that stresses bacteria will induce horizontal gene transfer. Mm-hmm. So personally, I think that the really key thing that drives antibiotic resistance development is selection, not the further triggering of more mutations, of more horizontal gene transfer. Because it's actually after our use of antibiotics that we got the explosion of antibiotic resistance as well. So I wonder, have you in any of your experiments looked at the potential for selection by these other compounds and not just increases of mutations and horizontal gene transfer?
Thank you very much for your excellent question, Giacomi. So we did some experiment try to decouple the selection and the mutation. So we use the bacteriophage, we try to say, because mutation is a random process, right? So we did some control experiment, we can rule out the selection play a very, very minor role for this kind of experiment. We do have that evidence in one of the publications. We have sent up like a control line to rule out the selection experiment, contributions. So I think but I agree with you, but it's not just individual effects, like through the mutation or gene transfer, is combined the effects. They do have a selection, they also have a mutation, but which pathway play the major roles is still to be further validated. We don't have a quantitative data to distinguish two pathways. Yeah. Thank you for your question. I hope I addressed partially.
Thank you, Jianhua, for your talk. It was very interesting. I really like your work. Célia Manaya from Católica University in Portugal. My question refers to the bacteria perspective. I mean, do you think, because I think that in most of these studies you are using commercial strains, domesticated somehow in our labs. My question is, do you believe that once in the environment, all bacteria will behave identically? And do you think it is something that is related with the taxonomic group, I mean the species, or do you think that is something that is indeed common to different phylogenetic groups and different physiological properties of bacteria?
Yeah, thank you again for your excellent questions. Yes, we consider this phenomenon is bacterial species specific or not. So in majority of my work, we're using laboratory model strain, but we do have our environment relevant bacteria. Actually, my understanding is if this drug or chemical, if they are very harsh to bacteria, so probably they can select or trigger mutation, they can promote gene transfer. So no matter which bacteria, this principally speaking, but the dosage will be different because some bacteria are very tolerant for example, for the drugs. Some bacteria are very sensitive. So that's why drug dosage will be different, or bacteria dependent. But principally speaking, if a drug are very harsh enough, they can promote no matter heavy metals, other nanomaterials, and even other drugs. They do have one. So that's why we did the active sludge. We did animal gaunt. So, but for that gene transfer, they do have some preferred or favorable bacteria will become trans conjugates. It's not all of them, depending on phylogenetic relationship. Yeah.
So yes, I'm Nicole Ricker. I'm from the University of Guelph, and it is my great pleasure to introduce our next speaker. This is Kaho Misawa from Kitasato University, and she'll be speaking to us today about the dynamics of Class 1 integrants in wastewater treatment plants.
I'm Kaho Misawa from Kitasato University, graduate of School of Medical Sciences, Japan. I will talk about dynamics of Class 1 integrants in wastewater treatment plants and hospital wastewater. Antimicrobial resistance has been recognized as one of the major global health threats. The annual number of worldwide deaths due to antimicrobial resistance bacteria infections was 1.27 million in 2019 and will increase to 10 million by 2050 if urgent countermeasures are not taken. Water environments are recognized as one of the reservoirs and transmission routes for the dissemination of antibiotic resistance, and integrants has been used as an indicator for antibiotic resistance determinants. Integrants allow for the acquisition and expression of antibiotic resistance gene cassettes used as indicators of antibiotic resistance determinants. There are 3 classes of integrants have been found, with class 1 integrants being the most frequently found in clinical and environmental settings. Class 1 integron integrase gene is proxy for anthropogenic pollution and has positive associations with the abundance of ARGs in wastewater treatment plants. The structure of a class 1 integron integrase is shown in the figure. Integrase recognized the ATTC in the gene of the gene cassette and incorporate and can incorporate the gene cassette into ATTI. This allows for resistance for antibiotics if ARGs are present in the gene cassette. Conventional detection methods have used the class 1 integron integrase gene to estimate the presence of ARGs. However, not all integrons may contain ARGs, and therefore, using class 1 integrase as an indicator may overestimate the presence of ARGs. In order to assess the true risk of antibiotic resistance, it is necessary to evaluate antibiotic resistance genes of human impacted integrons. In this study, we conducted experiments using a previously developed assay to determine the presence or absence of human impacted integrons. Integrons impacted by anthropogenic factors and carrying gene cassettes were defined as AINT1, and those impacted by anthropogenic factors but not carrying gene cassettes were defined as EAINT1. AINT1 targets the interface between ATTC in the gene cassette and QACE delta 1. And EAINT1 targets ATTI and ATTC. Each type was measured and evaluated individually. The aim of this study is to evaluate the dynamics of human impacted class 1 integrons with and without gene cassettes in municipal wastewater treatment plant and hospital wastewater, which is important in estimating the actual presence and dynamics of ARGs. Wastewater samples were collected 3 times from plant A and 2 times from plant B. Wastewater sample types were influent, secondary effluent, and final effluent. And hospital wastewater samples were collected over a 15-month period. DNA in the water environment can be classified into intracellular DNA and extracellular DNA. In this study, integrons in both iDNA and eDNA functions of municipal wastewater treatment plants and hospital wastewater were quantified. To do so, 50 milliliter of samples was filtrated using 0.22 micromembrane filter. iDNA was extracted from filter paper and was quantified using qPCR. eDNA was recovered by ethanol precipitation and was quantified by droplet digital PCR. These are the results of iDNA analysis into treatment plants. The vertical axis shows log concentration, and the horizontal axis shows from left to right influent, secondary effluent, and final effluent. Following the wastewater treatment process, INTI1 concentration decreased about 3 log copies per liter. In case of AINT1, and EAINT1, their concentration decreased about 2 log copies per liter. Both wastewater treatment plants showed similar trends. These results suggest that AINT1 remained more than 7 log copies per liter and released into the water environment. This slide shows the same result on a different sampling day. Together with the previous slide, the concentration of INTI1 was 1.4 to 11.4 times higher than the concentration of AINT1, suggesting a potential overestimation of the presence of ARGs using INTI1 as an indicator. Attention should be paid to the occurrence of new ARB by conjugation because of high concentration of ARB containing integrons discharged to the environment. Since similar trends were observed at plant A and B, further the experiments were conducted focusing only on plant A. The left figure is iDNA, and on the right is eDNA concentration. Target concentration in eDNA were about 4 to log copies per liter lower than iDNA concentrations. Interestingly, integron concentrations in eDNA increased from secondary effluent to final effluent. This is because chlorination has damaged the cells and released DNA. This DNA may contribute to increased ARB due to transformation. This slide shows the results of iDNA concentrations in hospital wastewater. Concentrations of all targets except for EAINT1 remained almost unchanged throughout the 15-month period. On the other hand, EAINT1 concentration fluctuated. The concentration of INTI1 was 1.2 7.5 times higher than the concentration of AINT1. AINT1 was always detected at higher concentration than EAINT1. This box plot shows relative abundance of AINT1 in municipal wastewater and hospital wastewater. AINT1 relative abundance in hospital wastewater was higher than in municipal wastewater. It is thought that antibiotics and other chemicals in hospital wastewater may act as a selective pressure, and many bacteria carry ARGs. These are the conclusions. INTI1 concentration was 1.4 to 11.4 times higher than AINT1 concentration and indicated an overestimation. If we use INTI1 as an indicator for ARGs, although the concentrations of AINT1 decreased during the wastewater treatment process, it was clear that more than 7 log copies per liter of AINT1 as iDNA and 4 log copies per liter of AINT1 as eDNA were released into the water environment. For eDNA, the concentration of each gene increased in final effluent more than in secondary effluent. Selective pressure might be higher in hospital wastewater than in municipal wastewater. The efficiency of horizontal gene transfer of antibiotic resistance genes in the water environment needs to be evaluated. Thank you very much for your attention.
So we'll will invite Professor Guo to come up and give his students' presentation as well. Thank you.
So this talk is mainly about interaction between correlation disinfection and AMR. So any relation between them? I want to use 3 studies to give you some indication. So as we know, we do have a very diverse waterborne pathogen in our water system, such as water distribution pipelines, premise and plumbing systems. So for example, we have Legionella, NTM, Pseudomonas aeruginosa, PO1, etc. They cause some disease through the water exposure. For example, if you do the shower, somehow they have aerosol. If you uptake that contaminated aerosol, you got the infection if your immune system is not strong enough. Okay, so that's why correlation is very important, or disinfection technology are very important to do the bacterial suppression. Or we need to mitigate bacteria or pathogen, make sure our water is safe to drink. So that's why we apply the UV oscillation or correlation very, very commonly in our water treatment, no matter for the drinking water treatment or plumbing system, and even recreation water, swimming pool, etc. But the question here is, how about interaction between AMR and disinfection? So how about AMR evolution even for the OPs under exposure long-term disinfection? So how about gene transfer even in the biofilm? Because so in our water distribution pipeline, we do have a biofilm attachment on the surface of the pipeline. So that's why so our gene transfer in biofilm rather than planktonic cells. So quite a few questions we want to address here. Okay, so first study I want to give you is about OP detection in the plumbing systems due to the COVID epidemic lockdown period. So back to a few years ago, we do have a pandemic. Many buildings were closed, right? So no matter in the office building or university campus building, they closed. So that's why you can imagine free calorie or residual calorie concentration will decay very, very quickly to be the low level or zero level or very, very low level free calorie. So that's why we were wondering, so maybe due to the long-term water stagnation, we have more waterborne pathogen growing in the biofilm. Is that true or not? So we did a study. What we did here is we collect samples from the 8 buildings in the UQ, the campus. So we have 4 buildings from the urban catchment, A, B, C, D. We have 4 buildings from rural catchment, E, F, G, H. For A and E, because some researchers are doing very critical research, so that's why the buildings still are open. So that's why they have water stagnation is not that serious. But the building B, C, F, G, so we closed that building for 3 months or even 6 months. For another 2 buildings, D and H, they even have a longer water stagnation. So we try to detect waterborne pathogen, including Legionella, NTM, and Pseudomonas aeruginosa PO1. So we're using a multiple method, so including flow cytometry, qPCR, and country dependent method, plus second sequencing to confirm which bacteria we have in such system. Okay, let's have a look at the results. So based on PCR, actually, NTM can be very frequently detected in such stagnated water. So more importantly, so this is a building D with very long water stagnation. You can see they harbor the highest NTM abundance, so compared to no other buildings. But this trend was not observed in the rural catchment building, because for the rural catchment, so free calorie is almost very, very low level. So that's why they have very high NTM as a baseline already. Even you have longer water stagnation, they didn't increase further. So this is so we also detect the Legionella and Pseudomonas in this building environment. So then we run the country plate experiment. You can see a lot of very beautiful morphology here. Actually, they are very alloying bacteria, NTM. We did a sequencing, we confirmed we do have some clinically relevant pathogen, and we also have environmentally relevant NTM species in such plumbing system. Okay, this is the first study, but I want to highlight here is this infection is very important because you can cure or you can mediate waterborne pathogen. But the next question we want to test, okay, so on the long-term disinfection, are there any lective effects? Are they able to trigger AMR or not. So that's why we did such like a mutation or evolution experiment. So design is very similar as I did show before. So we did a parasite experiment. We choose like three bacterial model bacteria, including E. coli, Legionella pneumonia, and Pseudomonas PO1. So we have multiple lines. We do that like the parasite experiment. So then we pick up cloning to scan like MAC and confirm mutation frequency through this equation. We also did like a DNA sequencing and RNA sequencing to confirm. So what gene mutation happened and how about RNA expression regulation. So this is results. So this is E. coli. You can say other exposure like sub-MAC free cholerae, so they can also promote like the mutation. So the high concentration, the higher mutation ratio, and the quicker like mutation happened in the E. coli. Very similar to the Pseudomonas PO1 and Legionella, even like up to 100-fold change in terms of mutation ratio. So then we rather than pick up like the cloning to do the MAC actually again. So for the free cholerae, they can select multiple drug resistant bacteria. So not only resistant one antibiotics, but multiple drug resistance. So the small question here is how about the acquired resistance can be maintained very, very stably or not? So we did like a further experiment. So what we did here is we remove the free cholerae stress. We do the passage without the free cholerae. You can say we still can maintain the resistant population like a fraction very, very stable. Even they have same like amount for resistant bacteria like a population. So for the fitness, actually after like the long-term evolution, the fitting cost didn't increase. Actually, they can maintain the fitness even higher. Okay, that means under such like a MAC for cholerae, you can select the multiple drug resistance, didn't increase the fitting cost actually for that resistance. Okay, so we did a further DNA sequencing. We can identify quite many like the genetic mutation SNPs. For example, we have like a stress response gene has been mutant. We also have a metabolism-related gene also be changed. Okay, we did like RNA sequencing. What we found, some like oxidative stress gene-related gene has been upregulated. So here we propose. So on the exposure for cholerae, they can also trigger SOS and oxidative stress. So they can even like upregulated efflux pump. So then and the transporters. So if you have antibiotics, they can pump up again to become the multi-drug resistance. Okay, last study is gene transfer in the biofilm. Because in the water system, all the water pathogen, like the colonize in the biofilm. So no matter is a plumbing system or water distribution system, so that's why here we develop a biofilm model. So we use the 1960 virus to develop like biofilm. So then once we have a very thick like biofilm, we spike the E. coli in the biofilm. We also add disinfectant agents. So then we want to say, can we quantify the gene transfer frequency in biofilm matrix? So here we have a 3 biofilm model. One is the E. coli biofilm. One is the Pseudomonas putida biofilm. So one is the multiple species biofilm. So we try to do underexposure disinfection. How about the frequency in this gene transfer? So this is results. For the upper panel figure is the control. So red here is a donor. And actually black here, you can't see that, is a biofilm recipient, is a received biofilm. We can't see it because it's black. But for that gray here, they are trace conjugates. For the control, just very few bacteria become trace conjugates, but under exposure, this like disinfection drugs, we have more and more trace conjugates like trapped in the biofilm matrix. So we did like quantification. Actually, under exposure 5 ppm or 10 ppm free cholerae, so conjugation frequency from E. coli to E. coli has been dramatically increased. But for the Pseudomonas, like we need a high concentration because Pseudomonas is more resistant to free cholerae. So that's why 20 ppm can increase like the conjugation for putida, but 20 ppm free cholerae will suppress like a conjugation because too harsh for bacteria E. coli. So this conclusion, I don't want to read it through again. So what I want to highlight here, actually, disinfection is kind of trade-off. It's double-edged like a sword. They do have a positive rules because they can kill bacteria. But somehow, if you don't use them very properly, they can also trigger AMR mutation. Last but not least, I would acknowledge my team members. So this work was conducted by stagnation conducted by Casey, and Zhigang did the mutation experiment. So Yujie did like a biofilm experiment. I also want to thank all of industry partners and collaborators and funding agency. Thank you so much.
Thank you for your presentation. My name is Nada. I'm a PhD student at McGill University. So my question, do you suspect that if we give antioxidant, it can decrease the level of horizontal gene transfer or not?
Yes.
Thanks.
For this, work, we didn't add antioxidant, but for some experiment, if you combine your drug, like a chemical and some antioxidant chemical, they can slow down, they can decrease mutation and gene transfer frequency.
So there is no other factors that can affect the horizontal gene transfer at this point with the chlorine treatment?
So
just the oxidative stress, it will be if we give the antioxidants, so now it's improved. So no other factor affect the horizontal gene transfer at this point?
Yes, once you add antioxidant, but what we saw is the gene transfer frequency will drop, but other side effects we didn't test yet.
Yeah. Okay. Thank you so much.
Yeah, but it's an indication here, for example, back to the first study, Gant and microbiota, right? Someone, if you, because one of my friends, his daughter got the disease, they uptake the carbamazepine very often because Gant and microbiota will be lost the balance. So that's why, like a doctor, they offered you can eat some yogurt or probiotics. Even someone mentioned you can eat some antioxidant, but I'm not sure in clinical applications over there. But it's a very good question. Yeah. Thank you.
And our third invited talk for this session is Kadir Yannick, who I'll invite up to the stage. And he will be speaking to us today about the role of bacteriophage as vectors of AMR in wastewater treatment plants.
Hi, I'm Kadir Yannick, and I'm a PhD student at the University of Manitoba. And I'm a little bit excited, of course. And I chose a dark background because I'm not a dark person, but it is just a tribute to the viral dark matter and all viruses who are in dark and undiscovered, unclassified. And I call it the curious case of the bacteriophages because on one hand, we know that they are actually weapons against some of the antibiotic-resistant bacterial infections, and on the other hand, there is some speculations and also there are some findings that they can be significant vehicles of antibiotic-resistant genes. So, and we know that in this era of the antibiotic-resistant crisis, we know that they are coming back. I mean, the phage therapy is making a comeback. And we know that it has also some other biotechnological applications. And it is also a proven concept that they can transfer antibiotic-resistant genes between bacteria by transduction. And but when I was doing my research, before I started my research, and what I see in the literature, there are lots of contradictory reports. Some says that they are highly abundant, they carry antibiotic-resistant genes, and those resistant genes in phages are highly abundant. But on the other hand, there are some other reports which says they rarely carry rarely carry the antibiotic-resistant genes. So I just want to briefly talk about what is happening here, and I want to talk about the critical steps in identifying, assessing antibiotic-resistant genes in in phages from environmental samples. And one of the most critical steps, I think, for me, it is the concentration of phage particles from very complex environmental samples. So those samples are very dirty, and still we are improving our methodologies to separate phages from the rest of the organisms in the environmental samples. So, and the other thing is the detection methods. One of them is the qPCR, and which will give us like an absolute quantification, and the other one is metagenomics, which will give us more information about what's going on there. So, and yeah, what I see in qPCR studies, qPCR-based studies, I saw that, like most studies, report a high abundance of antibiotic-resistant genes, and some of those studies actually do not really mention about what is the contamination of 16S or bacterial DNA in those phage samples. And when I look at the metagenomics samples, and there are two sides. One of them is the conservative IRG search, and the other one is the loose IRG search. And if you keep your IRG search criteria strict, and then you detect less IRGs. So I was curious. I was just like a innocent PhD student at the beginning, but I turned into a highly skeptical PhD candidate at the end because of this. So if they are carrying resistant genes, and if it is not overestimated, then we are in trouble because they are highly abundant. They are the most abundant entities in the wastewater treatment plants. So in order to assess this, I collect samples from the wastewater treatment plants, and I applied both qPCR and the metagenomics, and also in order to compare what is happening in bacteria and in phage samples, I did those studies both for bacteria and phages, and I collected samples from the influent, from the primary sludge, from return activated sludge, secondary effluent, final effluent, and also the sludge cake. And I collected them from 2 different wastewater treatment plants in 2001, in March and October. So this is one of the common methods that is used to separate like the bacteriophages from the environmental samples. What you do, you use like the 0.2 micrometer filter papers, assuming that, based on assumption that most of the phages are actually smaller than 0.2 micrometer, and they will pass through the filter, and the bacteria will retain on the filter. So I did this filtration step, and then I used skim milk flocculation, which is a method that I actually proved that it is working. And in my study about the COVID-19, I compared two different viral concentration methods, and this skimmed milk flocculation method kind of gave me the best results, and it is very cheap. And after these steps, I did nucleic acid extraction for the bacterial fraction. The bacterial fractions are the microorganism on the filters, and also in the filtrate, I concentrate the viruses, and I add DNase A and RNase to remove any bacterial DNA. And also I did the nucleic acid extraction. Then I also did RNase 3 to remove any residual 16S. I know that those methods are designed to minimize bacterial contamination. I have 2 fractions here, as I said. One of them is the bacterial fraction on the top on the filter, and the other one is in the filtrate, which is the phage fraction. And I expect that most of the DNA in the bacterial fraction would be bacteria and some phage. And in the phage fraction, I was expecting to be mostly phage DNA and a little bit bacterial DNA. So I targeted some resistant genes there, and these resistant genes are actually are reported to be carried by bacteriophages, and also I did the Illumina sequencing with the on NovaSeq platform. So, and these are some numbers about my contexts, which are determined as virals. In total, I had over 1 million of contexts, and then only around 400,000 of them were identified as viral, but we still don't know. Maybe we have more viruses because we don't know actually. It's totally in dark, and we don't know if there are more because we were only able to detect this many viral contexts. Maybe there are more. And also I clustered them, and after clustering, I had like 260,000 viral contexts. And most of them, like almost half of them, were bigger than 2 kilo base pairs, and around 10,000 of them were bigger than 10,000 kilobases. And also I had 154 complete phage contexts, which are like the complete phage genomes, and the 600 high quality, and the rest are the are medium quality, or the contexts with at least one viral context. And then I went into the classification. I'm talking about this classification is just to make a comment about how rare they are, actually, how it is hard to identify them, to classify them. So at the class level, actually, I classified most of them, and most of them are Codoviricidas. But at the family level, especially after the ICTV changed the viral classification system, I detected like only 15% of them at the family level. So, and also another thing that I saw, there is no significant difference between samples. There is no like a trend in terms of alpha diversity between samples. Like the diversity in influent is kind of very close to actually diversity in the effluent. It doesn't really change much. But when I look at the beta diversity between the wastewater treatment plants, actually, both wastewater treatment plants are quite distinct from each other. They are very different, and also, I think it's a good news because we have high diversity. It means that we might cover a lot of different bacteriophages. So, and this is my my analysis regarding to the IRGs in bacterial fraction. And as I expected, there is high diversity in influent samples, and there is less diversity in effluent samples. And also between wastewater treatment plants, again, there is more. So there is more. Both wastewater treatment plants differ from each other, but when we look at the time, actually time doesn't have a big impact on the diversity. So, and here everything looks normal. It is the results from the bacterial fraction, and in the bacterial fraction, the usual suspects are there. I mean, and most of the resistant genes are the sulfonamides, beta-lactams, multidrugs, and the lincosamides. And when you look at the host, they are Pseudomonas, Echeverria, and they are always the usual suspects there. And also we know that it is mostly mediated by mobile genetic elements. And I was only identify one prophage, which is carrying an antibiotic-resistant gene. So this is the results from the phage fraction. I only detected one resistant gene in the phage fraction, and which is like a complete phage genome, and the host is a Streptococcus, and it is carrying a lincosamide resistant gene. and with high confidence. So, and also I detected a prophage. Again, it's carrying a lincosamide-resistant gene, and it is also infecting Streptococcus. I thought it is interesting, but I don't know. I still don't know much about it. I don't know why it is the lincosamide-resistant genes and bacteriophages infecting the Streptococcus. So my IRG search criteria was strict for bacteriophages. So I lower my search criteria to identity of 30% and the e-value of 10 to minus 3 and the coverage of like 40%. So in this case, I detected 600 resistant genes in phage fraction. But I detected 600, and also I eliminated the DFR1A gene because they are highly similar to another protein in phages, which is a reported study. And at the end, I only detected 18 potential IRGs in bacteriophages. But what happened with the rest? And when we look at the rest, they are actually other the phage proteins. They are more similar to phage proteins. For example, the fluoroquinolones resistant genes are very similar to phage pentapeptide proteins. And when we look at, for example, vancomycin resistant genes, they are actually very more similar to phage tail proteins. So, and this is my quantification here. And again, everything on the bacterial side looks normal because it is decreasing from influent to effluent. And when I look at the bacteriophage side, when I don't detect 16S, generally there is no resistant genes. It's just like when there is bacterial contamination actually in the samples, then we detect more resistant genes. And also everything here, again, in terms of like the frequency looks normal, and it is very, since I don't have much time, I will go a little bit fast. Everything looks normal with the literature, but I have another table here which shows I also conduct a viral enrichment study, like how my viruses are enriched in my samples. And when I look at them, the less enrichment results in more IRGs. And when I look at it here, the highly enriched samples are actually result in very few IRGs here. So, and also I detected all the resistant genes which are detected by qPCR in the phage samples as well, phage fraction, but they were all plasmid or bacteria related. They weren't like phage related. And this is also my viral enrichment score is actually correlates with the, it is negatively correlates with QPs with abundance of antibiotic resistant genes. Here we see the results from the qPCR study and all of the QP and all of the IRGs and the 16S is positively correlating with each other, and at the same time the results from the metagenomics studies, the 16S and the IRGs in the metagenomic side of the story correlates with each other, positively correlates with each other. And these are my conclusion. I was only detected 3 IRGs, and here I said if for the assessment of the IRGs in bacteriophages, we have to be more careful. And also the limitations of these studies, again, is the viral concentration method, and also the depth of the sequencing. Thank you. Yeah.